Okay, I think we're good to get started. Thank you for coming to the 18th Annual Needham Healthcare Conference. My name is Emma, and I'm here to introduce Evelo Biosciences. We have Jonathan Poole and Stefan Riley.
Thank you very much, and thanks to Needham for inviting us to your conference, and good afternoon to everyone here in the room and listening on the web. My name is Jonathan Poole, Evelo's CFO, and I'm delighted to be here this morning representing my colleagues at Evelo. Evelo Biosciences is pioneering a new class of oral biologic medicines designed to modulate systemic immunology by targeting immune cells in the small intestine. I'm going to define what they are and how they could potentially transform the treatment of many major diseases. Importantly, we're already in the clinic, and we're on track to have as many as 10 clinical readouts over the course of the remainder of 2019 and 2020. Before getting into that, I'll remind you that I will be making forward-looking statements during this presentation. So Evelo is harnessing a new understanding of immunology to potentially enable more effective, safe, and convenient therapies to treat all stages of major chronic diseases and cancer. We call these oral therapies monoclonal microbials, single microbial strains that impact clinically validated pathways of disease by engaging with immune cells in the small intestine. Evelo was founded around a central area of evolutionary biology that has largely been ignored to date in the development of medicines, the fact that the small intestine is a central modulator of the immune system, the immune system's primary window, or portal, if you will, to the outside world. Immune cells in the small intestine have evolved to sense and respond to the environment. The nexus of the small intestine and our monoclonal microbials is critical to understand. It's the interactions going on here that initiate the cascade of systemic immune modulation, which has demonstrated such profound effects for us preclinically in our monoclonal microbials. The small and large intestines are different in many ways, as depicted on this slide. The small intestine has a very small proportion, around 0.01% of the total gut microbial biomass. It represents approximately 80 to 90% of the gut surface area, around the size of a tennis court. And the microbes which do exist in the small intestine are largely distinct from those in the large intestine. The staining on these micrographs highlights some of these differences between the small intestine on the left and the large intestine on the right. In these micrographs, you see the gut wall stained in blue, you see the mucus layer stained in green, and microbes stained in red. You see on the left, in the small intestine, the diffuse mucus layer, which enables a close association between the red microbes and the immune cells in the blue gut wall. This enables microimmune cell interactions, and by contrast, the large intestine has a thick mucus layer, as you can see in green on the panel on the right, which prevents this kind of interaction to the greatest extent. These characteristics enable our monoclonal microbials, dosed at the right level, to become the dominant signal acting directly on immune cells in the small intestine and thereby modulating immunity throughout the body. On slide 5, we talk a little bit more deeply about the mechanism of action of our monoclonal microbials. And this diagram shows that they commandeer a three-step evolutionary process to exert their multifaceted effect on disease. If you focus on the panel in the middle of this diagram, you see a cross-section through the wall of the small intestine. We depict monoclonal microbials as the orange circles on the inside of the small intestine. And the first step of the mechanism of action is the sampling by immune cells. In this case, we've indicated dendritic cells sampling the contents of the small intestine. And we dose and formulate our monoclonal microbials to be the dominant thing that is sampled by immune cells in the small intestine as they pass through. And the interaction of monoclonal microbials with these immune cells has the effect of conditioning the immune cells in a specific way for different monoclonal microbials, and we select that depending on the disease that we're targeting. These conditioned immune cells then circulate via the lymphatic system and condition T cells and other immune cells, firstly in lymph nodes proximal to the small intestine, and then by the lymphatic system they migrate to sites of disease to exert their effect. So remember, all we're doing is hijacking with our monoclonal microbials a key control mechanism of our immune system for therapeutic effect. And slide 6 demonstrates this in a little bit more detail with some data. And this is from a product candidate called EDP-1867, which is a recent preclinical candidate nomination from our research pipeline. And these charts represent different readouts from a preclinical delayed type hypersensitivity model. And in all of these charts, read them as the vehicle untreated cohort in black, a cohort treated with dexamethasone, one of the most effective anti-inflammatories that we know of, and then EDP-1867, our monoclonal microbial, depicted in blue. So as you can see here, at first glance, the big picture here is that EDP-1867 is having at least as good an effect on reducing inflammation as dexamethasone. That's quite remarkable for a therapy that's acting on immune cells in the small intestine to drive this effect systemically. Reading this slide in a little bit more detail, we can actually map the pathway of interactions of monoclonal microbials in the small intestine affecting immunology at the site of disease. So in the left-hand side of this chart, we see cytokine reduction of IL-5 and IL-6, both inflammatory cytokines, and we see 1867 reducing the levels of those cytokines at the lymph nodes proximal to the small intestine. And then we move into the middle of the slide, and we see a similar effect on a different panel of cytokines, this time occurring at the skin-draining lymph nodes, so proximal to the site of disease. Then the far right diagram here is the ultimate effect on disease, in this case, a reduction in the swelling of the ear. It's also important to understand that we can define the pharmacology of our monoclonal microbials in detail, and moreover, that pharmacology depends on their structure, dose, and formulation. On the left-hand panel of this slide, we've got another preclinical DTH model, and this time we're showing the effect of EDP-1066, which is one of our clinical candidates currently in phase 1b trials, versus vehicle and dexamethasone as our positive control. The green groups here on this slide represent different doses of EDP-1066. And so what you can take from this slide are three things. First, and quite remarkably, with EDP-1066, delivered in the small intestine, we're demonstrating a systemic anti-inflammatory effect equivalent to that of dexamethasone. Secondly, the effect is dose-dependent. And thirdly, parenthetically, we're dosing this with our current clinical material so we can manufacture at GMP scale and maintain efficacy versus our early research. Once again, EDP-1066 is getting nowhere near the ear in this model. It's exerting its systemic effect by the interaction it's having with immune cells in the small intestine. And data from this kind of model was what really first opened our eyes to the potential of monoclonal microbials. And since then, we've continued to work to understand these effects. And now we conclude that this efficacy is likely a property of their physical structure, and that dose dependency is likely driven by the magnitude of target cell engagement in the small intestine. Given these findings, dose and formulation optimization remain core to our research and clinical development strategies as we pioneer the development of monoclonal microbials as a new class of medicine. One of the key reasons that we've been able to advance our therapies into the clinic so quickly over the short course of our life as a company is the fact that we brought together a team with a rare combination of skills to develop a platform that can identify individual strains of microbes with the pharmacology to impact disease. And I'm using the word strain here to mean something like the difference between individual humans, but in bacterial terms. Their pharmacology is that specific, defined, and dose-dependent. On the left of this chart, you see that our process starts with a proprietary library of microbial strains, which we then interrogate through a series of in vitro assays with human cells and in vivo models of disease. We're looking for concordance of the effects of our monoclonal microbials across these assays in order to identify potential candidates for clinical development. And we've also built highly valuable capabilities in process development and manufacturing and in formulation to allow the monoclonal microbials to be delivered to the small intestine. Our current formulation is a convenient daily oral capsule designed to protect the monoclonal microbial from stomach acid and release in the small intestine. We're aiming to deliver the right dose at the right time, at the right place for the right effect. Let me reinforce here that these medicines drive integrated effects on multiple pathways, and that means we've got the potential to impact diseases in ways not addressed by single target therapies. These profound systemic effects come without systemic exposure, so we do not see colonization or a need for metabolic activity in our monoclonal microbials. The target is the immune cells in the small intestine, and the monoclonal microbials otherwise pass normally through the digestive tract. We've borrowed the term monoclonal from the antibody field to reflect that our GMP manufacturing process always starts from a single clonal strain in a proprietary master cell bank. That enables us to reproduce efficacy reliably as we move through development and scale towards commercialization, and we anticipate that commercial manufacturing will be cost effective, which will enable us to have significant global pricing flexibility. And we think this unique profile drives the potential for monoclonal microbials to transform the treatment of many diseases. Imagine the potential of an orally delivered, well-tolerated daily medicine with efficacy akin to the current standard of care therapies. On slide 10, we show the progress we've made in a very short period of time, the 3 or 4 years since we've been around as a company. It's been quite remarkable. We've been incredibly productive, and we now expect 10 diversified clinical readouts across 2019 and 2020. I'll review the specific clinical trials in more detail later in the presentation, but the highlights here are that we have 2 therapeutic areas currently in clinical trials, inflammation in blue, oncology in red. In inflammation, we have 2 monoclonal microbials currently in phase 1b trials in atopic dermatitis and psoriasis. We're expecting the first data for EDP-1066 later this quarter, and then in the second half, data from EDP-1815. This is the start of our inflammation disease portfolio, and we expect to advance into other inflammatory disease indications later this year. In oncology, we have EDP-1503 in 2 active clinical trials across a basket of 5 arms of different oncology patients, ranging from microsatellite colorectal cancer patients to triple-negative breast cancer, basket of anti-PD-1 relapse patients, and then naive and relapsed melanoma patients. All of these studies are being done in combination with Keytruda, our own study in a clinical collaboration with Merck, and the melanoma study is actually an investigator-sponsored study being conducted by the University of Chicago. So a very broad pipeline, a lot to look forward to over the coming rest of 2019 and 2020. Let me now review our inflammatory programs in more detail. Firstly, our initial clinical trials, and then I'll give you an insight into our longer-term inflammatory disease portfolio strategy. As I said, we have 2 ongoing clinical trials in psoriasis and atopic dermatitis for EDP-1066 and EDP-1815. The primary endpoints of these placebo-controlled trials are safety and tolerability in healthy volunteers and patients. Importantly, we're also studying exploratory endpoints which will give us insight into the clinical efficacy and immunological effects that our monoclonal microbials are having in patients. We're treating patients for 28 days with daily EDP-1066 or EDP-1815 at 2 different doses, and these doses are scaled based on estimates from our animal models at 1 times and 5 times the effective dose in preclinical models. For EDP-1066, we announced in February that we successfully completed dosing of the healthy volunteer cohorts in that study, and that we advanced into dosing of patients on plan, and we're expecting initial clinical data from this study later this quarter, as I mentioned. For EDP-1815, this trial continues to enroll on plan, and we expect to announce initial clinical data from this trial in the second half of 2019. And we see a significant opportunity to address the needs of millions of patients with atopic dermatitis and psoriasis, driven by the unique potential of monoclonal microbials. A convenient, safe and well-tolerated oral medicine with efficacy potentially akin to current standards of care therapies. And specifically, this opens up the potential to target the large, moderate or pre-biologic patient population who lack treatment options at present, either because they're uncontrolled or non-compliant on low-dose steroids, or they're ineligible or averse to injectable biologics at the other extreme of the disease spectrum. Now, along with the attractiveness of these opportunities from a commercial perspective, we selected atopic dermatitis and psoriasis as our first clinical trials because they'll enable us to conduct, firstly, relatively quick trials, but secondly, very informative trials. As I mentioned, we're taking punch biopsies of the skin, and we hope to be able to elucidate from those punch biopsies is the pharmacology of our monoclonal microbials, and that may lead us into different clusters of inflammatory disease in the future. So based on what we see in these trials, we may take one product, for instance, into psoriatic diseases if we see movements in IL-17 or TNF-alpha biology, or indeed another into the atopies if we're seeing movements in IL-4, 5, or IL-13. Ultimately, we envisage a portfolio of monoclonal microbials for different inflammatory diseases, and we see EDP-1056 and EDP-1815 as the start of that portfolio. As I mentioned before, we've already nominated EDP-1867 as our next anti-inflammatory monoclonal microbial. Shifting gears to oncology, where we're looking for pro-inflammatory characteristics in our clinical candidates. EDP-1503 is a special monoclonal microbial which we found through a comprehensive screening effort to select for the strongest potential anti-tumor profile. We've shown that EDP-1503 has a truly pleiotropic effect on many anti-tumor immune mechanisms, an ability to multitask and modulate multiple immune pathways at once. So the big picture here, as you look at this textbook range of clinically validated mechanisms of tumor immune stimulation, is that we see potential for 1503 to be complementary to checkpoint inhibitors. As they remove the brake on the immune system, EDP-1503 may be an effective way to broadly activate the immune system or hit the accelerator. One of the most striking preclinical observations relates to MHC class I upregulation, which we show in the chart at 10 o'clock on this slide. This points to EDP-1503's potential for use in cancers which downregulate MHC class I expression in the cells, meaning that there are no antigens being presented for the immune system to respond to. Here we see that EDP-1503 in red increased significantly induction of MHC class I over the control. So this is an exciting observation, and it's driven our clinical strategy to partly focus on cancers such as microsatellite stable colorectal carcinoma, which has no good treatment options at present and is unresponsive to checkpoint inhibitors. So the potential for us to make a difference here is obviously very exciting for us. As I mentioned, we're currently studying EDP-1503 in two separate open-label clinical trials. The first study is our own study. It's a clinical collaboration with Merck. And patients here are dosed daily with EDP-1503 monotherapy for a period of 2 weeks, and then they receive daily EDP-1503 alongside KEYTRUDA thereafter. And so we're looking at the effects of combination therapy in microsatellite stable colorectal carcinoma, triple-negative breast cancer, and a basket of PD-1 relapsed patients, with the potential with our monotherapy run-in to understand some of the immunology that we're driving independent of that combination therapy. Our guidance for this trial is that we're announcing clinical data, initial clinical data in the first half of next year. Our second oncology clinical study is an investigator-sponsored study being conducted by the University of Chicago, this time in melanoma, in both naive patients and patients who've relapsed following initial checkpoint therapy. And we expect initial clinical data from that trial in the second half of next year. So building a strong foundation for Evelo with our initial focus on inflammatory disease and oncology in the clinic. And on the one hand, we see these therapeutic areas as our core. On the other hand, as depicted on this slide, we're only scratching the surface of the potential of our platform and monoclonal microbials more broadly. So not only are we advancing the candidates in research and development through information and oncology applications, but we're also generating very interesting data pointing to the potential for us to apply our technology in metabolic and cardiovascular disease, neuroinflammation and neurodegeneration, and a little bit further over the horizon, the potential in autoimmune, neuropsychiatric, and vaccine applications. And importantly, we're executing on plan in an important year for Evelo, and we have the potential to show a clinical signal and to demonstrate proof of concept of our platform and monoclonal microbials as a new class of medicine. Our 3 current product candidates alone are expected to deliver 10 clinical readouts in 2019 and 2020. And the first of these readouts, as I said, is for EP1066 in atopic dermatitis and psoriasis, which we expect this quarter, followed by EDP1815 data later this year. As CFO, I'm happy to remind you that we're well financed heading into this important period for Evelo, with around $148 million in the bank at the end of 2018. So the potential to demonstrate proof of concept of these products in these diseases is exciting in its own right. But for us, the opportunity to validate the potential of monoclonal microbials as a new modality of medicine speaks to the ambition of Evelo, opening up the true potential of the platform as we seek to bring new therapies to large patient populations around the world. With that, thank you very much for your attention, and I'd be happy to take any questions that you may have.
So, you're giving an oral drug to get to the skin, I guess, going through the intestine, and like, what exactly is your platform?
So the question is, what exactly is our platform that enables us to deliver an oral therapy targeting immune cells in the small intestine and yet have a systemic immunological impact on, in this case, the skin in relation to atopic dermatitis and psoriasis? So what our platform enables us to do is, from a library of a microbial library, we can select for strains with defined pharmacology, and we do that by taking individual strains and putting them into a series of assays involving human immune cells. So we co-culture single strains of microbes with human macrophages and dendritic cells to see how the microbes change the phenotype of those immune cells. We also do a series of animal in vivo models, and we're looking for concordance between what we see immunologically in the human cell systems and both the immunology and the disease effects we're seeing in the animal models. And then from that, we nominate clinical candidates. We go through a process development and manufacturing step and ultimately advance those candidates into the clinic. And we've done that successfully so far with our 3 programs that are in the clinic.
So having an anti-inflammatory effect in the skin, but would that be causing a side effect somewhere else in the body because it is going systemically?
Yes. So interestingly, we've not seen that to date in our animal models. We are obviously in our first phase 1b trial right now. As I mentioned, we've successfully moved through our healthy volunteer cohorts and had that data reviewed by the Safety Review Committee. Generally, in animals, we've not seen any dose-limiting toxicity at the doses that we've explored. And again, we're having these systemic effects without systemic exposure, in the sense that the monoclonal microbials themselves are interacting with immune cells in the small intestine and then just passing through normally in the digestive tract.
These are alive?
So the question was whether or not the monoclonal microbials are alive. No, the answer is that they don't need to be alive. So one of the things that I mentioned in the presentation was that I think we've concluded that the mechanism of action for monoclonal microbials is a structure-function reaction. So we think it's structural motifs. And the way in which we elucidated that is actually we've experimented with intentional steps in the manufacturing process to render the monoclonal microbials non-replicating, and so essentially dead.
So essentially that's why you do daily dose as well?
Yes, I mean, so right now we're dosing, daily dosing in the trial. That's been the regimen we've used in preclinical models, and ultimately over the course of clinical development we'll determine whether or not that's ultimately necessary in the long run.
How have you been doing? You said you have a lot of cash, so
Yes, so we were funded through our life as a private company by Flagship and a number of other private investors, and we IPO'd last May. So we're now listed on NASDAQ, and we raise money from a series of high-class public institutions. Okay, thank you very much.
I'll let you get on stage.
Okay, good idea.
Very cool. I have two.